Welcome. I will present to you today our recent work on synthetic extracellular matrix, or ECM. To create our synthetic ECM, we use Phycocolloids and therefore are translating seaweed materials from the sea to the bench. Hello, my name is Dr. Aurelien from the University of Freiburg in Germany. Thank you for joining. Our work ultimately will address the shortcomings of organ transplantation. As you know, it will be never possible to provide to every patient in need of organ transplantation with a functioning, compatible replacement organ. Recent technologies are using cell therapy and try to offer this technology to repair defect organ by using autologous cells. The cells from the patient are harvested, then reprogrammed, and finally reintroduced into the patient to repair the defect organ. We are material scientists and we aim at using biomaterials that carry specific information to direct the repair of organs using learnings from the natural extracellular matrix. Our hypothesis is that we can use materials to direct the cell fate and function by creating a morphospace that recapitulates early developmental process. A linear view of development suggests that the relationship between genes and the process they affect would involve a linear causal chain where genes affect key parameters of development which ultimately will tune morphogenesis. An integrated view of development considers the effect of genes on cell properties that due to cell-cell interaction influence tissue geometry and then feedback to gene expression pattern where the extracellular matrix is playing a key role in shaping the development of organs, which could be harnessed to create materials able to repair injured or defect organs. And so to create these materials and transforming seaweed materials into useful biocompatible hydrogels, we use the 5 elements of our Phycocolloid transformation roadmap, from the extraction of polysaccharides, then chemical modifications of this polysaccharide, finally understanding the structure property relationship using existing or developing new methods to process these polysaccharides into different aspects, hydrogels or fibers, and then finally developing biomedical applications. And so our research integrates upstream with Phycobiology Research Institute and also farming industry, and downstream with a company that transform this innovation into biomedical products. And so if we have a look at the natural extracellular matrix, we can see that it's composed of proteins and polysaccharides that form the mechanical structures of the tissue and of this environment. The cells in green here attach to these proteins and polysaccharides through cell receptors in yellow, and around it we have soluble signaling such as growth factors that move freely into these hydrogels. If we want to mimic the extracellular matrix, we can use hydrogels that do not carry any information, and we can introduce information such as using cell adhesion peptides to simulate the adhesion of the cells into onto the hydrogel, and this is done through the connection of the cells through cell receptors to the cell adhesion peptide and then to the hydrogels. In this hydrogel, like in the natural ECM, we can add soluble signals such as growth factors. And so using these models, we can answer key questions to understand what are the interesting information, what are the cues that we need to influence cell fate and function. So the first one is to look at the mechanics and try to understand which hydrogels stiffness and elasticity can drive cell organization. The next one is to use cell signalings, and so to look at these cell adhesion peptides that can connect the cells to these hydrogels. And finally, how these signals are presented, how the stiffness, how the biological signals are presented that can influence the cell organization, and so to look at patterns in these signals. And so first, in this talk, I will focus on the mechanics and show which work we have been conducting into trying to answer this question of the hydrogel stiffness and cell organization. As you know from mechanobiology, the stiffness of the environment can direct the fate and function of cells, and so it's been quite extensively demonstrated that a stiffer environment is better for cells that are evolving into a stiffer tissue. And so to recapitulate this sort of signals, using Phycocolloids, we try to understand what sort of chemical modification we can do to have a hydrogel that have tunable mechanical properties. And so we use Agarose, which is the chemical structure that you can see. And if we do oxidation of the primary alcohol into a carboxylic acids, we are able to modify the stiffness of the hydrogels. And so as we, with the same concentration of polysaccharide, when we found the hydrogels, if the higher the chemical modification, so the higher the concentration of COH in the polysaccharide chains, the lower the shear modulus is. And this, we know that how this is coming. It's because when we have these carboxylic acids, we disrupt the secondary structure of the hydrogel, and we have been validating this hypothesis by using molecular dynamics, as you can see here in the video. And so when we start with a double helix and we have carboxylic acid instead of primary alcohol, double the chance, then this double helix is then disrupted, as you can see here. And so as we move towards, then the video will see that then the two polymer chains are really not touching anymore each other. And so this was done with molecular dynamic, but then we can go further and try to look at the secondary structure using circular dichroism. In blue of this spectra, we can see the optical rotation of native agarose, and this is what is considered the pattern of a double helix. And if we do the same experiment with carboxylated agarose, we can see that we have a shift of this helix, and then the appearance of a new elbow in this spectra. So we attribute that to a change of the conformation of the polymer chain from an alpha helix to a beta sheet type of organization. And we can see here on the scanning electron microscope of freeze-dry hydrogels of native agarose, we have some sort of helical organization. But then if we look at the same hydrogels, but processed hydrogel formed with carboxylated agarose, you can see we have this sheet organization, so showing that the helices are then disrupted. And so this, now that we have these materials, we can change the mechanical properties by just changing the amount of carboxylic acids groups on the backbone. So we can form these hydrogels and use it for vasculogenesis models. And so what we did is that we, with this carboxylated function, then we can react cell adhesion peptides, so this RGDSP sequence that binds to integrins, and we can chemically bind this peptide to the carboxylic acids functional groups of the modified agarose, and then the endothelial cells can bind through cell receptor to integrins onto this RGDSP. And we found out that when we supply this gel with specific mechanical properties with the right growth factors, we can force endothelial cells to form this luminal organization, which is typical for endothelial cells. And this construct that can be matured then in vitro and then form this structure of a couple of hundred of micrometers height. So in vitro, we identified a specific formulation, so specific cell adhesion peptide and specific mechanical properties to drive the organization of endothelial cells into some sort of blood vessel-like structure. And so to validate this, we moved then in vivo into a model of angiogenesis. And so we injected this RGDSP functionalized carboxylate agarose into the muscle of a mouse, and we looked at the result after 7 weeks. We found out, so we tried different stiffness, stiff and soft hydrogels, and we see here in the muscles on the H&E staining that the hydrogels remain inside the muscles, and in the mesenterychum we can see that we don't have any formation of capsule around this hydrogel. So the hydrogel is well tolerated by the mouse, and what we found out is that when we looked at this what type of cells and the close look to these hydrogels, we found out that the stiff hydrogels with RGD and without growth factors gave us this sort of organization of cells. But then when we looked at the soft hydrogels, so the same type, same stiffness as the formulation we use in vitro, we had the formation of a dense vascular network, and also this without growth factors. So we were able to identify a formulation in vitro and then move this formulation in vivo to show that to identify cues of the extracellular matrix based on stiffness and cell adhesion peptide to formulate a hydrogel that is injectable and can drive the formation of new blood vessels without the use of growth factors. Next, we wanted to look at the cell signaling. So we knew that RGDSP was really good for angiogenesis, but the next question we wanted to know is that what is the role of the cell adhesion peptide? And so the first step was to look at the literature and to look at what sort of peptide sequence have been used in tissue engineering and synthetic extracellular matrix. And so we found out that the majority of paper report the use of the RGD sequence for the patterning or for the functionalization of scaffold, hydrogel, fibers, or even implants. We have other cell adhesion peptide like IKVAV and YIGSR that are less used. And so when we calculate the percentage of the ratio of each sequence, we can see that 89% of the publication at the time were reporting the use of RGD. And the remaining 6% for AKVAV, 4% for YRGSR. And so our question was, can we go beyond RGD and can we use other one? Can we extend this library? Of course, RGD is really interesting because we know it is water soluble, which is really good for processing and functionalization of polysaccharides and other polymer. And also it binds to integrin, which is present in many types of cells. And so we try to look at what sort of cell adhesion peptide we could, if we could extend this library of use and find new cell adhesion peptide that could be relevant for biomedical applications. And so the medical application that we targeted was spinal cord repair, and to see if when you have a cord lesion, many scientists have shown that you can inject olfactic uncheating cells to repair this defect. However, once these cells are injected, we want them to remain in place and to fasten the healing of this spinal cord lesion. And so to do that, we want to identify a hydrogel that could be used to drive the organization of these olfactic uncheating cells and the better integration of these cells into the spinal cord lesion. And so to do that, to develop this as a study, we used synthetic hydrogel, a PEG 4 arms that is functionalized at the end with divinyl sulfone, and another one which is functionalized with thiol. And so we can use a reaction that pairs the vinyl sulfone with thiol at room temperature under physiological pH. And what is good with this is that we can also integrate within the 3D hydrogel network. We can integrate peptides that are terminated with thiol. So we can synthesize a library of different peptides with at the end a cysteine amino acid. So when we mix these two polymers, we can create hydrogels, and here this is how we can see the formation of this hydrogel. So this is the in blue, this is the G prime and G double prime of the hydrogel, so the PEG vinyl sulfone with the PEG thiol, and in red this is the PEG vinyl sulfone with the PEG thiol and with mercaptoethanol as a simulation of the peptide. And so we can see that when we have a peptide, it takes a bit more time to form the gel. But the gel with the mercaptoethanol, so with the peptide, it's a bit lower in stiffness, but still form a strong gel. What is good with this system is that we can automatize the screening of cell adhesion peptide, and we can automatize the formation of this cellular environment. And so what we did is that we use this PEG 4-arm with thiol, PEG 4-arms with vinyl sulfone, then the cell adhesion peptide, so a peptide with the cysteine at the end, and the cells, so these olfactory uncheating cells, and we use a liquid handling robot. So the robot will first pipette these PEG 4-arms thiol into a 384 well plate, and then we form a premix of vinyl sulfone, cell adhesion peptide, and olfactory uncheating cells. We mix them together, and then finally we add them to the thiol terminated PEG to form a hydrogel. And so to using this automatized handling of liquids, we were able to screen 16 different cell adhesion peptides and to look at how these olfactory uncheating cells were organizing these hydrogels. So on tissue culture plastics, the OECs are organizing like these cells on two-dimensional planar surface. In PEG, when we don't have any cell adhesion peptide, they remain round, so they do not adhere, they do not spread. So this is our negative control. And of course, with RGD, we have this linear organization of these cells, so they spread, and they attach nicely to the RGD peptide sequence through integrin adhesion. And so we look at different peptides, so the IKVIV and other ones, and we found out that these two ones, CCAR RSR and CPR-ARI, were forcing the cells to organize from this linear organization. So that we think could be used to direct the repair of spinal cord lesion, because they use as these OSCs then guides the lesion repair. So when we looked a bit deeper into this, the effect of this cell adhesion peptide on the OSCs, we found out that we didn't have any change of the phenotype of the cells in all these different cellular environments. Also, we found out that these cell adhesion peptides were also driving the proliferation of these cells to a higher extent than tissue culture plastic. And the big difference between tissue culture plastic and these peptides, hydrogels, that we are in 3D in the hydrogel, so we can have way more cells growing in a bigger volume, of course. And so, of course, in PEG, we have few cells proliferating, most likely at the bottom of the well. And so we think that this screening platform can be used to extend our library of cell adhesion peptide, because we can now identify quickly with the automatized system new adhesion peptides. And that we could use that short term to identify new peptide for the functionalization of surfaces such as wound dressing, implant, but also fibers. In the midterm, to use this platform to identify new cellular environment to simulate disease. And finally, on the long term, to create multicellular and multitissular object using 3D printing. So now that we have a technique, so we have a materials to look at the mechanical properties of cells, we have also methods to look at the cell adhesion. So what sort of cell adhesion peptide we can use for specific type of cells. We were interested in looking at the cell signal presentation. So what sort of a cue or pattern of the signals can we use to direct the cell organization? And so the biggest difficulty that we have was to develop a method to create patterns inside these hydrogels. And this is what I'm going to show you today, how we created patterns and gradient environment into this synthetic extracellular matrix. So this study, or this journey, started by looking at how we can process our carboxylated agarose with 3D printing. And the first step, or the first method we used, was a droplet 3D printer. So we have a liquid cooled stage. We have agarose here as a melted soft hydrogels, and we can extrude it through a macro valve to deposit droplets. And as soon as the droplet reached the hot droplet of agarose reached the cold stage, then we have a transition from the solution to a gel, and we can form a 3D object. However, when we have a flow through the when we extrude this hydrogel with the cells inside, we might generate stress to the cells and even death to these cells. And so for us, it was really interesting to see what sort of hydrogel formulation can we use for this 3D printing method to limit the cell death and limit the shear stress during extrusion. And so this is where carboxylated agarose play a big role, is that with the same concentration of hydrogel, we can have different elastic modulus, so which means that we don't change the amount of cells, so we do not change the shear property of the solution during extrusion, and we can change then the mechanical property of the resulting hydrogels. And so we extruded different formulation, so native agarose, 28% carboxylated, 60% and 93% carboxylated, and we can see that the native agarose, which is a really strong hydrogels, we had also way more death of cells with this FDAPI staining, and this we think this is correlated through the shear stress inside during extrusion. As you can see here, so during extrusion, the CA93 has low shear stress and a low dead cells by field of view. And so with this technique, we were able to create different patterns and have cells inside this 3D-dimensional object. We were also able to 3D print human mesenchymal stem cells inside this pattern. So we have a soft hydrogel embedded inside a stiff hydrogel. So the interest with that is that we can organize cells in a specific environment inside another type of environment. And so ultimately the goal will be to print different cells, each in their own mechanical environment. However, with this technique, we can pattern the object with different type of cells in different environments, but the transition from soft to stiff is really net. It's a clear cut between the soft hydrogel and the stiff hydrogel. So we don't have any gradient, and so the environment that we have is in which the cells are deposited is the one that they will see. So we don't have any smooth transition between the soft and the stiff hydrogels. So we wanted to look at how we can develop a printing process where we could have gradients and different patterns. So for that, we developed a new bioprinting setup where we have two extrusion pumps. So we have the hydrogel inside this syringe, and then we have these two syringe pump. The hydrogel is kept warm above the gelling temperature into these blankets, and then we have the mixing head where the material A from the syringe from the left, mix with the material B from the syringe from the right, and then they are assembled inside the mixing head and then extruded and then cooled on to these cooling plates where the gel, this carboxylated agarose gel, is setting up. So first we had to develop printing heads. So as I said, we have red material then mixed with another type of material, for instance. And so the issue is that when you want to mix two different materials inside a small needle, then you have the laminar flow that takes place, and so you don't have any mixing of these two materials. However, when you have this static mixer, then we are able to disrupt this laminar flow and having mixing of the materials. And so now we were able to 3D print an object with this setup. So we created this ring of carboxylated agarose, and to be able to create this environment. So this is the object we decided to study, and we decided to add some gradient of stiffness, gradient of cells, and gradient of immobilized signals. So why do we want to have this sort of gradient? Is because we know that if we want to have a design of materials, and if we want to have organization of the cells, we want to simulate, we want to give some cues to these cells. And so our approach is to have a design of the materials where you have different patterns. Then these hydrogels are biofabricated with cells, and the cues on the material then direct the organization of the cells following these patterns. And then we have maturation of the cells according to this pattern of signaling. So what type of pattern of signaling do we have, or do we know? And then for that we looked at developmental biology, where we have 3 types of patterning. So the first one is chemotaxis, which is the gradient of soluble signals that can drive cells movement and cells organization in vivo. We have also mechanotaxis, which is the gradient of mechanical properties, which also has been shown to direct cell movements and cell organization. And finally, haptotaxis, which is a gradient of immobilized signals, most of the time cell adhesion signals. And so here, what we want to do is create a gradient of cell adhesion peptides inside our hydrogel matrix to guide the cellular organization. And so how can we create a gradient of mechanical properties? So we have shown that we can mix the native agarose with the carboxylated agarose, and by doing that, by varying the ratio of carboxylated agarose inside the hydrogel made of native agarose, we can change the mechanical properties in the same way as when we just chemically modify the native agarose. And so if we know that we could then use our mixing head, printing head, to introduce different amount of carboxylated agarose to a solution of native agarose. And so the process is that we have a stiff hydrogel that we mix with a soft hydrogel inside the printer head, and then as we extrude the hydrogel, we can form a pattern for mechanotaxis and to create a gradient of mechanical properties. And so this looks like this. So as we print, we add the soft hydrogel, which is here labeled in green. So we have this one. So we did the same with the blue dye as well. So as we move through this through the object, we have the addition of the soft hydrogels, and then we measure with the through indentation. We looked at the modulus in compression. So as we move higher into the hydrogels, which is 10 millimeter high, we have a softer and softer object because we have added more and more soft hydrogel. So now that we have this mixing head, we can also maybe do some chemical reaction inside the mixing head. And so for that, we modified our carboxylated agarose with maleamide functional group through EDC coupling. So now we have a soft hydrogel with a maleamide functionalized functional group. That we can see here on the enema, we have the apparition of a double bond here. And so now we can react this maleamide with a peptide that is terminated with a thiol through a reaction that is extremely quick. As you can see here, we have the kinetic that we have studied through UV visible light by looking at the disappearance of the color from the maleamide. And so in a couple of minutes, we have the formation of the covalent bond between the maleamide and the thiol. So now our mixing strategy is to have the native agarose with the maleamide soft hydrogels, and then we can add as well a peptide that is terminated with a thiol to create our peptide, cell adhesion peptide functionalized agarose. And this enables us to create a gradient of cell adhesion peptide through apoptosis. So the mixing is like this, so that we mix, we add different concentration of RGD peptide that is tagged with a FITC dye so that we can see it through UVVs and also by eye as we print. And so in 2D, we can see that we can increase the concentration of this peptide, but also in 3D here on this hydrogel. So as we go through the layers, we can increase the concentration of this cell adhesion peptide. We can do the cells to create the same with cells to create a gradient of cells. So here the cell solution, the cell media is supplemented with phenol red, so we can follow the addition of this cell solution into the hydrogel. And so in 2D, we have more and more cells present into this two-dimensional organization of this two-dimensional printing, but also in 3D we can do the same. And so the idea here would be to have cells that can generate chemotaxis, so that the cells will generate the soluble signals inside the three-dimensional object to create a gradient of soluble signals generated by the cells and to recreate this chemotaxis. So now we have this in 3D. We have also, as we go through this object, we have more and more cells inside, and we can calculate the number of cells inside each layer, and also we know that then these cells are still alive. And so the idea would be to create this pattern of material that can guide cells migration and cell organization in vitro, but also ultimately to do a printing in vivo and then recruit cells into this hydrogel with a specific pattern. And so this concludes our talk on this synthetic extracellular matrix based on Phycocolloid agarose and modified Phycocolloid carboxylated agarose. I would like also to showcase other work. So if you are teaching polymer chemistry, you can have a look at our website to acquire a card game to teach your students free radical polymerization. And also recently I have published a textbook on biofabrication by Doug Ritter, so that recapitulates all the methods and concepts of today's field of biofabrication. Thank you for your time. Don't hesitate to contact us and to follow us on Twitter or Instagram, and also to visit our website www.forgetlab.com and to maybe order your free radical polymerization card game. Thank you for listening. Bye bye.